I welcome you to this video on initial proof of DNA as genetic material. Frederick Griffith in 1928 demonstrated the transformation of the non-pathogenic R strain of Streptococcus pneumoniae into the pathogenic S strain by the heat-killed S strain. He could not identify the chemical nature in the heat-killed S strain causing the transformation. However, he called such a chemical as transforming principles. Oswald Avery in 1944 identified the chemical nature of transforming principle, providing the first evidence that DNA, not protein or RNA, is the genetic material that is responsible for the transformation. In this video, I will explain the basic principle, methodology, key conclusion, and criticism of the groundbreaking experiment and findings of Oswald Avery. So what could be the chemical nature of the transforming principles that might have caused the transformation in Griffith's experiment? Avery et al. To address that tested the following three biomolecules in the transforming principle causing the transformation. The first one was protein, second DNA, third RNA. These biomolecules were hydrolyzed by specific enzymes and individually tested for their ability to transform the R strain into S strain in vitro. Avery's experiment was the first direct evidence that DNA carries genetic information. How he did? He took heat-killed S stain, homogenized it and filtered it. So he used filtrate. Filtrate was divided into three tubes. They were treated respectively with the indicated enzymes, RNAs, protease and DNAs. The filtrate treated with these enzymes were individually added to the flask containing R stain, and then they identified the S strain in the grown culture. In flask number 1, which was treated with RNAs, transformation was identified because S strain, both S and R strain were there. Same was the case when the filtrate was treated with the protease. Both R and S strain were identified, suggesting transformation. So these findings suggested that protein and RNA are not the genetic material. In flask number 3, interestingly, there was no S strain identified. So only no S strain was identified, only R strain was there, indicating absence of transformation. These findings therefore suggest that DNA may be transforming factor causing the transformation of R strain into pathogenic S strain. So this is the first experiment, elegant experiment, that suggested that DNA is the genetic material. However, his findings were extremely criticized. Scientists were skeptical about DNA being the genetic material because of its simplicity. They thought that DNA is made up of only 4 nucleotides, whereas protein is a better candidate because it is a more complex, made up of 420 amino acids. Therefore, it lack universal acceptance. Moreover, the protein centric inheritance was very dominant at that time. Therefore, they also raised the possibility that there may be protein contamination in his preparation. Furthermore, there was no direct evidence in higher organism at that time. Therefore, his work was criticized. So thank you very much for watching this video. I request you to like it, to share it and to subscribe it. Until next time, you take care. Thank you. Bye bye.